Good afternoon, dear students. Today we are going to study blunt end ligation, also under the heading of joining of DNA molecules, right? And how we are going to join the DNA molecules or to perform blunt end ligation. We perform blunt end ligation by using linkers, adapters and homopolymer tails. Okay, so blunt end ligation, joining of DNA molecule. Now see, yesterday we have discussed about restriction enzyme. What is the use of restriction enzyme? Restriction enzyme is used to cleave a DNA molecule. But restriction enzyme cleaves a DNA molecule at a particular recognition sequence which we call as which is a specific site, okay, consisting of 4 to 6 base pairs having palindromic sequence. But there are two major ways in which a restriction enzyme cleaves a DNA, breaks a DNA, either creating blunt ends or sticky ends. Now what are blunt ends and what are sticky ends? Sometimes restriction enzymes, for example like HIND2, cut the DNA in the middle of their recognition sequence. Okay, in the middle of their recognition sequence, leaving behind blunt ended DNA fragment. Right. Now these fragments which are having blunt ends do not have tendency to stick together. They do not stick up or attach easily with another DNA molecule. Right. The T4 DNA ligase enzyme cannot ligate or join such blunt ended DNA molecule very efficiently. Their ligation occurs by chance only. Further, very high concentration of DNA ligase is as well as DNA fragments are required when the DNA to be joined have blunt ends. Right. Now what is that blunt end? If you look at this DNA molecule, okay, this is a double-stranded DNA, okay, having base pairs joined to each other, right? Now, what happens if a restriction enzyme cleaves the DNA? Okay, suppose for example, the restriction enzyme cleaves the DNA in between like this. Okay, what will happen at that time? The DNA will be converted into two DNA fragments having same 5' to 3' end. Okay, that means their ends are blunt. Their ends are blunt. Like you can see here, this is a DNA molecule having the base pairs which is shown by a blue mark. These are the base pairs, right? Now, so the cleavage. What you can see that both the DNA fragments that are formed here are having same double strands. The two strands are same. Okay, so this is called as a blunt ended DNA. So restriction enzyme cleaves like you can see here. The restriction enzyme has acted upon the recognition sequence is CCC, and the enzyme SMA1 has acted upon it and it has formed two DNA fragments, both of which are having same length. Okay, their 5' to 3' ends are equal. Such DNA fragments are called as blunt ended DNA molecules, right? So there are certain restriction enzymes which create blunt ends. And these blunt ends are not desirable in recombinant DNA technology or in genetic engineering experiments because T4 DNA ligase cannot join such blunt ended DNA molecule with a foreign DNA. Okay. Further, the concentration of DNA ligase required as well as blunt ended DNA required is too high and they join only by chance. So what do we require in recombinant DNA technology experiment? We require DNA fragments having sticky ends. Sticky ends are also called as cohesive ends. Okay, now what are these sticky ends? These sticky ends are single stranded extended region at the 5' end. Okay, we can see here the blue dotted line is showing the double stranded DNA which is acted upon by enzyme here and here, restriction enzymes. Okay, so this will give you two fragments of DNA, but each fragment is having a single stranded extended arm. Okay, it is having a single stranded extended arm. This single stranded region is called as sticky ends. Okay, so these single stranded 5' overhangs, right, they are also called as 5' overhangs or sticky ends or single stranded regions which are generated due to the action of restriction endonuclease, right? So what is desirable? What kind of DNA fragments are desirable in genetic engineering? Those having the sticky ends or cohesive ends, right? You can see 5' phosphate group. So at the 5' end they have extended single stranded region which is called as sticky end or cohesive end or 5' overhangs. What are they called as? 5' overhangs or sticky ends, right? Now, so you can see here from this diagram, you can see BAM H1 has acted upon the DNA at the particular recognition sequence, and you can see here extended regions at the 5' end. So and they are single stranded regions. Such single stranded regions are sticky ends or cohesive ends or 5' overhangs, very much desirable in genetic engineering experiments. Like you can see here, this is the sticky end, okay, that is present in the one molecule. Say for example foreign DNA fragment to be cloned, okay, and this is the one, okay. Here the red one is the vector, and the green one which is showing having sticky end or extended arm is the foreign DNA fragment. You can see that because there are single stranded extended ends, right, the two molecule, the red one which is the vector and the green one that is the foreign DNA fragment, fit into each other easily because they are having single stranded extended arms having complementary base pairs. So fitting, or you can say joining, is very efficient. T4 DNA ligase brings about joining of fragments very efficiently if they have sticky or cohesive ends, as you can see here. How do they get fitted? Very easily, right? Now, but because of the action of restriction enzymes, sometimes blunt-ended DNA fragments are created and these fragments are needed. Okay, so what we can do is, we have to attach the sticky ends to this blunt-ended DNA. What we have to do? That is what is called as blunt-end ligation. Blunt-end ligation means what? Ligating a sticky end, joining a sticky end to the blunt ends of the DNA, right? In such situation, when restriction enzyme produces blunt-ended DNA fragment, the recombination frequency lowers because of unavailability of suitable site on the DNA to be manipulated. Now, sticky or cohesive ends are absent on the blunt-ended DNA fragment. These cleavage sites can be added externally. Okay, we have to add them. How do we add them? By using linkers, adapters or homopolymer tails. Okay, so blunt end ligation or joining of blunt ends with the linker, adapter or homopolymer tail will convert the blunt end DNA into a cohesive end or sticky ended DNA. Now what are linkers? See, linkers are chemically synthesized. What are linkers? Linkers are chemically synthesized, okay, double-stranded small oligonucleotide sequences. Okay, linkers are chemically synthesized, artificially synthesized small double-stranded DNA oligonucleotide sequences containing one or more restriction enzyme site. That means they are small double-stranded DNA fragments. Simple. Linkers are small double-stranded DNA fragments which are chemically synthesized. In these linkers, site for restriction enzymes are present. These linkers are joined to the blunt-ended DNA and then they are treated with restriction enzyme. So restriction enzyme will cleave at the site present on the linker and convert it into cohesive ends or sticky ends, right? So the linkers are small chemically synthesized double-stranded DNA molecules attached to the blunt-ended DNA. They have restriction enzyme site for cleavage by restriction enzymes like EcoR1 or HIND3 or BAM-H1, like that. Linkers are ligated to the blunt end of the DNA using DNA ligase, right? Now both the vector as well as the foreign DNA to be cloned, the vector DNA and the foreign DNA to be cloned, are then treated with the restriction enzymes to develop sticky ends. The sticky or staggered cuts are then ligated with T4 DNA ligase. Okay, so we have a blunt-ended DNA. It is mixed with linker attached to the blunt-ended DNA molecule. Then the blunt-ended DNA attached with linker and the vector both are treated with restriction enzyme. The restriction enzyme will cleave at its side and develop sticky and cohesive ends. Now when you mix the foreign DNA fragment with sticky ends with the vector, they will join each other. So the staggered ends or sticky ends are then ligated by T4 DNA ligase with very high efficiency, and recombinant plasmid DNA or recombinant vector DNA molecules are produced. Okay, so you can see here in this diagram, right? We have a foreign DNA fragment, double-stranded DNA having 5' to 3' end. The ends are blunt. You can see both the strands are having equal length at 5' end also. So this is a blunt-ended DNA. Now these blue, small double-stranded DNA molecules are linkers, right? So now the linkers are having recognition sequence in between. As you can see here, this linker is having a recognition sequence GGA-TTC. So the BAM H1 enzyme will cleave the linker at this site, okay, and generate sticky ends. Now the blunt ended DNA shown in red color is joined to the linker DNA, okay, which is shown in blue color, and then it is treated with BAM H1 enzyme. And when it is treated with BAM H1 enzyme, the linkers have recognition site for restriction enzyme. So there the restriction enzyme will cleave the linker, and as you can see here, 5' single stranded ends are generated, which are called as cohesive ends or sticky ends. Similarly, the vector molecule. Suppose this is a vector molecule, okay, that means it is a plasmid DNA, okay. This plasmid DNA is also treated with BAM H1 enzyme. Now when it is treated with BAM H1 enzyme, again you can say cohesive ends will be generated in the vector molecule also. And now this molecule, that means the foreign DNA having sticky ends and the vector are mixed together, and T4 DNA ligase will insert the foreign DNA fragment into the vector very efficient, like that. Okay, so this is how linkers help us in converting the blunt-ended DNA into sticky ends. Now here we have shown both adapters as well as linkers. You can see that. Now I'll show you this diagram next. First, let us understand now what are adapters. What are adapters? Adapters are also short double-stranded oligonucleotide DNA strands having pre-formed sticky or cohesive ends. What are linkers? Linkers are chemically synthesized small double-stranded oligonucleotide sequences, double-stranded DNA fragments. They have site for restriction enzyme, right? But what are adapters? Adapters are again short double-stranded DNA molecule, but they have pre-formed sticky ends or cohesive ends. They already have sticky ends. There is no need of treating them with restriction enzyme. Okay, why do we use adapters, or when do we use adapters? We use adapters when restriction enzyme has site within the foreign DNA to be cloned. That means this was your foreign DNA fragment and this was attached with a linker. Okay. Now the linker is treated with restriction enzyme so that sticky ends are generated. Simple, right? So the linker is treated, sticky ends are generated. But the restriction enzyme that we are using to convert the blunt-ended DNA or to generate sticky ends in the linker, the same restriction enzyme may have site within the DNA also, within the foreign DNA to be cloned, right? So in that case, what will happen? The foreign DNA to be cloned will be broken down by the restriction enzyme, okay? So when the restriction enzyme used for generating sticky ends in the foreign DNA in the linker has site within the foreign DNA also, then we do not use linkers. We prefer using adapters, right? So you can see here, this is a foreign DNA fragment shown in black color. Linkers attached to the foreign DNA fragment. Okay, linker and foreign DNA attached by T4 DNA ligase. And these are adapter molecules. They already have preformed sticky ends. So there is no need to treat them with restriction endonuclease. But where linkers are attached, we treat with BAM H1 and you have foreign DNA fragment with sticky ends. Now this is a plasmid DNA. Okay, this one is a plasmid DNA vector treated with restriction enzyme, and you can see here sticky ends are generated, and then the foreign DNA with sticky end. The foreign DNA with sticky end is mixed with the vector, and what you get is a recombinant vector. Okay. So for this purpose, we require foreign DNA fragment with sticky ends, right? So this is an adapter molecule where you can see that at the 5' end there is already a sticky end, preformed sticky end. We don't have to treat it with restriction enzyme. Now what are homopolymer tails? See, homopolymer tails are small oligonucleotide sequences, single stranded sequences having the same base. Like if I say poly A tail or oligo DA tail, means it is a small sequence consisting of A. So it's a small sequence having A. So it's called as poly A tail or poly DA tail. Okay, D stands for deoxyribonucleic acid, right? Now, so it is having poly A tail or poly T tail or oligo DA tail or oligo DT tail having A, TT sequences. These sequences, if the foreign DNA is having blunt end, then these sequences are added to the foreign DNA fragment to be cloned as well as to the vector. Okay, so we add the sequence, that is homopolymer tail, to the foreign DNA as well as to the vector by the action of enzyme terminal transferase. We have studied this enzyme in DNA manipulating enzyme. Terminal transferase enzyme helps to add poly A tail or poly T tail to the DNA molecule. Right. Now, how they are added? The homopolymer tails are used in such a way that if foreign DNA is to be added with poly A tail, that is oligo DA tail, then the vector will be added with poly T tail. Simple. If your foreign DNA, in your foreign DNA, by the action of terminal transferase, if you are adding poly A tail, you have to add poly T tail to the vector. So that the foreign DNA has A sequence and the vector has TT sequence. A and T pair with each other, and hence the foreign DNA will get easily fitted inside the vector DNA. Okay, so poly A tail and poly T tail develop on foreign DNA and vector respectively. So when they are mixed together, they unite to form covalently closed circular recombinant DNA molecule. Okay, so you can see here in this diagram, there is a small blunt ended DNA. We can see here the blunt ended DNA molecule. So we have added poly T tail here by the action of enzyme terminal transferase. Similarly, we have DNA fragment with poly T tail added. You have a plasmid treated with restriction enzyme, and then by the action of terminal transferase, what we have added? Poly A tail. Right? So the vector is having poly A tail and the foreign DNA to be cloned is having poly T tail. So when you mix them together, mix the two together and add T4 DNA ligase. Okay, add the T4 DNA ligase enzyme, and then the foreign DNA will get fitted into the vector DNA. Okay, and T and A will pair, and what you get here is called as a recombinant plasmid, because it is a plasmid DNA having a foreign DNA inserted inside it. Foreign DNA is inserted inside the foreign DNA is inserted inside the plasmid DNA, so it is called as a recombinant DNA. Now, if poly DT GC tails are used instead of poly AT tails, a high initiation temperature up to 37 degrees Celsius is required for the annealing reaction. Okay, the major advantage in using poly GC tail is that hybrids are more stable than AT hybrids. Okay, so when we use poly G and poly C tails, they are more stable as compared to, because 3 hydrogen bonds are present, and AT pairing is less stable. But still, homopolymer tails, homopolymer tails, linkers and adapters are used for converting the blunt ended DNA into sticky ended DNA, or for joining the sticky ends to the blunt ended DNA. So blunt end ligation is brought about by using linkers, adapters and homopolymer tails. So that is all about blunt end DNA ligation.